I think we can start. Yeah. I'm glad to have the privilege of moderating this interesting session. It's my pleasure to introduce Dr. Ahlström. I hope I don't miss too much the pronouns. Who will be talking about blastocyst selection, static versus dynamic morphological assessment. I invite the speakers to keep to the timeline to allow time for the debate at the end. Please.
Thank you. Okay, I'd like to thank the organizers firstly and Giovanni personally for inviting me to speak at this conference and to again visit Bologna. I was here for many years ago when Escher was here, and so I'm going to talk about blastocyst selection, and just to put a little bit of context, in the country where I work, in Sweden, we're not allowed to perform PGT-As, so many of the selection that we perform is in fact using non-invasive methods, and the ones that we test are using non-invasive methods. So blastocyst selection for us using either static or using dynamic has been really an interest area for us because we want to see a clinical effect in how we're performing IVF. So in my talk, I'm going to sort of evaluate whether it's just enough to make a few static observations in a short or very specific time intervals, or is it really beneficial to look at the full spectrum of developmental pathways an embryo takes before selecting it to transfer back to our patients. I hope to give you a little review of what's the status is right now, but also then look at what can really affect the reliability of both of these methods and look at some of the clinical evidence that's been published recently. A conflict of interest. One of our clinics actually has an agreement, a scientific agreement with Vitrolife, but in concerning with this talk, it has no effect on the results we're presenting. So there's a lot of images that we analyze within the laboratory, and I would be, as an embryologist, very biased to say that those that are the most important are the ones of our embryos that are developing in vitro. And that's because we're making very crucial and critical decisions by these static observations or by these dynamic observations when we're deciding the fate of each embryo. And we're always trying to improve the quality of these observations. We're always trying to make them a little bit more objective, a little bit more quantifiable, because we want to really decide and determine which ones have a clinical impact, because the rest is just noise. And we don't really need it to, in fact, select the blastocyst to transfer or to biopsy or to freeze. And the whole aim really is live birth, to select the blastocyst as fast as we can that can result in a live birth. So I think we're all familiar with Gardner score. I think it's well established in many clinics and internationally, and it's probably well established because it works quite well. We know from a lot of data in many of the clinics that, in fact, if you grade these three parameters, you can pretty much rank the order in which your blastocysts will implant from high to low chance. And if we do it at specific times, we get even more information about how to rank these embryos. And of course, we've set a threshold for good quality at 3 pp, but we know that that threshold is actually being lowered today. And that's from the work from Dean Moorbeck and even Danilo, who's, I'm not sure if he's here, but showing that borderline blastocysts also have a potential for our patients. If we're looking at what is really the consensus today and what do we know from the evidence, it suggests that a good trifectiform grade is in fact important for predicting live birth. And if we want to rank embryos further, developmental speed is important. And this is represented by your expansion grade, but also on the day in which your blastocyst expands. So a day 5 embryo or blastocyst, which has a discernible inner mass and a trifectoderm, in fact, has a higher live birth chance or an ongoing pregnancy chance than a day 6 and then a day 7. And this is applicable to frozen and fresh, to autologous and to donor blastocysts, but also to euploid blastocysts. Is there anything important in the stages leading up to blastocyst stage? I think we've debated this for quite a long time, and this is as long as I've been an embryologist. for over 2 decades, and there really is no consensus. In fact, and I only think it becomes very important when you have 2 identical blastocysts if you want to look back and reflect and perform static observations earlier. When it comes to time-lapse variables or dynamic variables, looking at the whole array of timings and the different sequences of events that occur during an embryo's development, there really is a lot we can look at and a lot to evaluate and still hasn't been totally evaluated. But we're able really to measure these timings of events and determine whether or not they're important. And we're also now to define new events, new morphological events we weren't able to see before. And there's the most interesting that have come lately are the cytoplasmic strings that are being also evaluated by Danilo and Laura Rienti's group, but also spontaneous collapse, which is also showing some sort of indication of whether or not you have a viable or non-viable embryo. But of course, these aren't really being assessed in selection models right now, so I won't continue the talk talking about these, and I'm sure Danilo will present something about these in the later talk tomorrow. So in consensus, is there a consensus about time-lapse variables? There really isn't. We really don't have one model that fits all laboratories. There's a few parameters that come up repetitively in the data and show that timing around start of blastulation or the duration of blastulation or the time of blastulation are important for selecting blastocysts for transfer. But no consensus is arrived yet today. I think we need to also remember that many of these morphokinetic variables are being assessed on top of a Gardner score. So you're already pre-selecting your embryos based on Gardner score, and then you're seeing whether or not you can fine-tune your selection by using these time-lapse variables. But I will show an example of a report that came out in which showed the reverse. They based their selection on time-lapse variables and then have assessed Gardner score. So how reliable are these? And they're reliable in different ways, and we need to be aware of this. How can we affect the reliability of these measurements that we're performing and when we're selecting our embryos? One of the most clear, apparently clear, sources of variation when we're looking at Gardner score is that at best within the same clinic with the same embryologist, you will in fact get at best moderate agreement between your embryologists. And it's also been shown that between clinics, this agreement for grading embryos according to Gardner score gets quite low and requires continual training to achieve a suitable level to know that an embryologist scoring a blastocyst in one laboratory is giving the same score as a blastocyst in another laboratory. And even on the individual level, if you look at the parameters, you can see that trophectoderm or expansion grade has a higher agreement than, say, grading the inner cell mass. So this will affect, in fact, the predictive ability of the Gardner score. When we're looking at the morphokinetics, it's actually a very good agreement. So when we're looking at the timing of events or the timing to start a blastocyst, many of the laboratories are very, very clear and achieve very high precision when actually defining the timing of these events occurring in an embryo's development pathway, which suggests that if there is a predictive value, that we've already really, really specifically defined and are on top when we say that this embryo is being defined, the T5 value is defined as a T5 value. Another thing that we've learned from the time-lapse field is really that, developmental grade is shifting, and it's not a static observation doesn't mean the embryo is at that stage the whole time. In fact, it changes over time. And so when we're performing these static observations and we're doing it at variables times of development on day 5, we're in fact cannot have the same prognostic value for this. And this is really nicely shown in this Australian study where they looked at the timings in which the Blastocyst score was being evaluated, and they saw there was a great variation. It was from 108 hours up to 128. So what they showed also in the multivariate logistic regression analysis was that if you didn't consider the hours post-insemination that you actually performed the Gardner score, this was a confounder of how well the Gardner score performed in predicting live birth. And so I think some studies that really don't hold themselves and report the timing on which they perform the Gardner score, in fact, you will find a variation in how this predicts live birth. For dynamic variables, there's another effect which is caused by the developmental timings, and this is really nicely shown by Marcus Messaguer's group. Often we see this nice histogram or normal distribution for different morphogenetic variables, and we're told that on the ends, in fact, that's where you see the timing difference. You see embryos which have low implantation potential, but that's not really the case. What you get, in fact, as being shown here, I think I can show this. You see in these histograms, and what you're seeing is an overlap of both the negative and the positive implantation or embryos that have implanted. And in fact, what we're trying to define is very, very small differences in these timing intervals for morphokinetic variables. And, you know, we're trying to see, is there a separation in these histograms? And can we use that to predict? And what we know from their work and what we know from other work of others is, in fact, these are affected by the culture conditions. They're affected by, if you're in one laboratory or in another laboratory, that the timings or the optimal timings shift. And you can see this quite clearly in the T5 here, where the quartile of timings is shifted. So one clinic is faster T5 value, whereas another clinic is slower, which can also affect the value of this in predicting in an algorithm whether or not a blastocyst should be selected. And as I mentioned, there's a lot of clinical confounders actually that can both impact the Gardner score but also impact these other timing variables. And I think Amy Barry's group has really demonstrated this very complex relationship when we're looking at how the way we culture embryos and how the patient and treatment characteristics can really impact morphokinetic variables and how they can be used for prediction or for selection of blastocysts. So what about the recent clinical evidence? So if we now put static observations up against morphokinetic or dynamic selection performance. This is a paper that's been published by Alice and Campbell, and they've done a lot of work on morphokinetic selection, and it's actually quite an interesting paper because here what they've shown is they're using an in-house classification model where it's only based on two morphokinetic variables, the timing to start of blastulation and timing of duration of blastulation. And what they show here, they rank their blastocysts from A to D, and they also do not use the Gardner score. They blind their embryologists to the embryo, what they call the embryo rank, and they ask the embryologist to score these according to Gardner score. So now we've taken away the foundation that we use in most of the studies, which is Gardner score, to first pre-select our population. But now they're actually just looking at the performance of their own selection model, and then we're trying to see if Gardner score can improve or is as well performing as this model. And what you can see in their adjusted AUC, in the black line, is that their embryo rank model actually does much better than the transfer rank model or the Gardner score model. But you see in the grey zone, this is the generalization error. It can sort of be in different populations. This may not, in fact, be the case. And what you can see also in the transfer rate, when we're looking at what sort of embryos were being evaluated or being transferred, that majority of them were actually a BB, a grade BB for Gardner score, which sort of suggests that this population of embryos may not have been selected in another clinic as the highest or the best quality. One of the algorithms that we've been working on is called KidScore. It's a part of Vitrolife. It's a commercial algorithm. It uses a number of different variables, early variables, and also the trifectodome and the cell masquerade to select blastocysts, or suggested to select blastocysts. It's been shown in the French groups and also in Japan by the Cato groups that, you know, if you first pre-select your blastocysts based on the Gardner score, and then you apply the IDA score, or the KidScore onto these blastocysts to see, you know, where are they ranking in the KidScore. And what you can see is that with increasing KidScore, there's an increasing in live birth or ongoing pregnancy rate. So it suggests that this KidScore could discriminate between embryos that were good quality, that would further discriminate them to say that, okay, good quality, and then put on the KidScore, and then you maybe can select better blastocysts to transfer. Well, we wanted to test this, and so we did a randomized controlled trial that was published last year, in which we first took patients that had at least 2 good quality blastocysts. We applied we selected embryos that were of good quality or of the highest good quality in the control group, but in the kid score group or the study group, we first pre-selected all blastocysts in which were of a grade 3B and above, and then we applied the kid score on top to then do the final selection. And we didn't really find in our study any improvement in the ongoing pregnancy rate using this algorithm. In fact, what you can say is they performed just as good as each other. Not just saying that you want improvement, but in fact, using this really didn't make a difference. And there was a lot of blastocysts in this cohort that in fact result in an ongoing pregnancy. And then we've moved on to AI, and AI has changing the ballgame because now we're taking the embryologist out of the picture and we're allowing the AI deep learning models to really make an analysis of the image. We're no longer manually inputting into what we consider the image to be of a good image or of a poor image. And this has been done both on static observations, but also being done on dynamic sequences of time-lapse sequences. And there are a number of commercial, which I'm sure you've seen, a number of commercial algorithms that are already present there for us to use. So if we look at some latest, I chose a recent study that was published looking and reflecting on the speaker before, talking about a strong association with blastocyst morphology and aneuploidy. This study actually wanted to use blastocyst or to use the static image to see how well can we predict aneuploidy versus euploidy, and then also attach on other components that we have available to us, like the patient's age, the morphokinetics, and also the blastocyst grade, and to see if you can create a supermodel that would really help us, and that would be beneficial for us in Sweden who can't do PGTA to predict aneuploidy versus euploidy. What they showed, or what my take-home message from this was that really the only great non-invasive marker that we have of aneuploidy for us to use is maternal age. And, you know, even if we added the morphokinetics to the image that was analysed by AI, it didn't have any effect on the accuracy of the model. The blastocyst grade, moderately to fair, maybe improves some points of accuracy. But if we really wanted to really get to the level at which we can get with PGTA, I have a little bit of reservation that blastocyst morphology, even when analysed by AI or dynamic variables analysed by AI, will in fact predict PGTA. But I hope to be proven wrong, because I would very like to have that method in our labs. We have also been working on an AI system as well called EDA. It's also a commercial algorithm that was supplied by Vitrolife. It was designed or developed in Australia by a group that really basically wanted to automate blastocyst selection. So they showed the AI or deep learning model all the images from the whole cohort and said, okay, first I want you to sort out which ones we can discard and which ones transferable, and then inside a transferable population also determine which ones will implant versus those that are implant. Whereas remember, the KID score or other algorithms usually are developed on embryos that are transferable. They report a nice AUC. Of course, when you're just looking at transferable embryos that are pre-selected, the AUC goes down. We applied this EDA algorithm onto our live birth data for our single blastocyst transfers. And what we saw, in fact, was that, yes, there is in those blastocysts that we've transferred that are resulted in, there's a higher live birth rate for those that have a higher EDA score, indicating a little bit of a potential to improve on our selection models. Although you can see by the width of the bars, the majority of those embryos that we're in fact transferring have already a high EDA score. And this is nicely shown in work by Danilo, which showed that, you know, EDA and the blastocyst morphology have some sort of relationship in where high EDA scores, even with the II, is giving a high EDA score to those embryos that have a high Gardner score quality. So, and this is even shown on the individual level when you're looking at inner cell mass or trophectodome, that you get higher EDA scores for high inner cell mass or high trophectodome grade and lower. Although there are these tails and there are these population of embryos at the end that show that you know, there may be some selection potential, in fact, where you can see embryos that we've given acceptable for transferring, but IDA would have deselected these from our population of transferring embryos. And we wanted to help our Australian friends, and so we contributed to a randomised controlled trial, and I'd hoped it would be finished or through the review process so I could show you the results of this, where we've actually applied IDA score into select blastocysts for transfer, but hopefully maybe on another occasion. But in this study, it was a non-inferiority. So what we're trying to determine whether or not IDA is just as good as Gardner score, basically, in selecting our embryos, and then maybe lighten the load or the workload or maybe more standardise more what we're doing in the laboratory. So as my last slide, and on time, I'd just like to state and maybe more isn't always more, and I think we really need to, more doesn't always make us make better decisions, and we really need to evaluate what we're bringing into our laboratories and do what we're doing, get the information, assess how important it is before we apply it, and I think we're doing a pretty good job of that, but we have varying discussions about how well it's going for each other. But I think what's being presented and what Giovanni will present soon is, we don't know everything yet, so we need to keep data mining and see how these different processes and time lapse can help us be better at what we need to do. So thank you.
Thank you very much for this really interesting and comprehensive overview on blastocyst selection. We will take questions after the next lesson. So it's my pleasure to call to the podium one of the scientific source of this meeting, Giovanni Coticchio. I think it doesn't need presentation.
Thank you very much. I'm very pleased to be here and be surrounded by so many friends and new colleagues. Today I will convey a message that I hope will complement nicely with the previous talk. And I'll try to share the notion with you that time-lapse technology, often combined with other technologies, allied technologies, can expand our understanding of oocyte, zygote, and embryo function and dysfunction, of course with a clinical twist, hopefully, in the end. I think we can all agree on the fact that movement, dynamism, attract attention, is very intriguing, like shown in this painting of Giacomo Balla, a futurist painter of the early 20th century. And in fact, embryo development by its very nature is very attractive from this point of view. Shape changes over time, sometimes in a very amazing fashion. And just giving you an example of how much information we can retrieve from that sort of data, of manifestation. These are the papers published by our group alone on fertilization, just to give you an idea of how much work we can do on that. And in fact, time-lapse technology can be further extended to previous stages compared to the beginning of life we try to achieve in an IVF lab, because it can also reveal developmental mechanisms which are relevant to the maturation of the fully grown oocyte. Here you can see a mouse oocyte which is going to extrude the polar body, the first polar body, and you can also see the chromosome pushed against the cortex. Now, it has been shown by time-lapse technology that streams of cytoplasmic particles assist this process of female meiosis. They push the metaphase 1 spindle against the cortex in preparation for the emission of the polar body and keep in place the newly formed metaphase 2 spindle in preparation for fertilization. But can we actually be objective in this sort of assessment in appreciating this cytoplasmic movement? Yes, we can. We have technologies to do that, computational imaging systems by which we can track and measure particle movement within the cytoplasm. And this can be expressed in numbers and also more visually with vectors with direction and, as you can see here, and color can express the type of movement and also its intensity. And over time, again, time-lapse technology, you can also appreciate, focusing on the red color of those vectors, that velocity is more is increased in the vicinity of the metaphase 2 spindle, just for the very reason I told you before. Now, we've tried a few years ago with Maurizio Zuccotti and other colleagues to harness this technology of analysis of cytoplasmic movements. And what we wanted to test was the hypothesis that by looking at those movements within the first 48 hours of development, we could predict blastocyst development through an entirely new approach. No time, no developments, no morphology, gross morphology, general morphology, but just movement of the cytoplasm. And in fact, what we did as well was to generate this particle image velocimetry data and feed them and feed an artificial intelligence platform in order to make some sense of those data. And in fact, although the study is pilot in its nature, it requires further expansion, we were able that this entirely new approach was able to predict blastocysts better than an experienced embryologist in terms of accuracy, sensitivity, and all the parameters you can appreciate in the slide. So there are different ways to skin a cat, if I'm allowed to say that. And we can actually, by time-lapse technology, access novel type of data and novel approaches to embryo analysis, also for clinical purposes. But basic observation is equally, I think, fascinating, and it can promise the possibility to develop new approaches for embryology. Now, in this paper, a group of scientists, including myself, were able to map a very detailed morphokinetic map of fertilization, which was missing until then. But it was not just a passive observation. There was a prediction made in this paper that those novel parameters could suggest alternative parameters of embryo viability. And in particular, there were a couple of those new manifestations, newly observed or newly described, that attract our attention: the fertilization cone and the cytoplasmic wave. That I'm going to show you what it is. The fertilization cone is something that appears immediately afterwards of the emission of the second polar body, and in standard IVF, not necessarily ICSI, represents a sort of a bump of the cortex of the oocyte. Presumably, this is an interpretation, induced by the male chromatin. It is a molecular interaction in which the DNA generates a distortion of the cytoskeleton of the cortex of the oocyte. On the contrary, the cytoplasmic wave is something that, after fertilization, generates a kind of a tidal wave of the cytoplasm, or again, cytoplasmic particles that go all the way across the oocyte cytoplasm. Now, the prediction in this paper was that the fertilization cone on the left-hand side and the cytoplasmic wave together could signal the initial position of the sperm DNA at the start of fertilization. So why I'm telling this story, apparently marginal? Because these and other morphokinetic phenomena may in fact reveal the origin of abnormally fertilized egg, opening to possible clinical use. And I'm going to tell you what I mean by that. There is a recent paper published by a Japanese group in which the authors observed what happens in presumably abnormally fertilized oocytes, 1 p.m. oocytes. In the upper sequence, you can observe what normally occurs in standard IVF. The sperm penetrate somewhere in the oocyte, the two pronuclei are formed, and they converge toward the center of the oocyte in preparation for the first cleavage. But there are other situations in which this egg fusion is spatial in a way, is peculiar. And that happens when this penetration of the spermatozoa occurs very near to the point where presumably the female chromosomes are located, which position is signaled by the extrusion of the second polar body. Now, therefore, the by looking at, again, the fertilization cone and the cytoplasmic wave, which should indicate the position of penetration of sperm, they were able to identify where actually sperm-egg fusion occurs. And by, of course, looking at several examples of this manifestation, they were able to conclude by morphokinetic analysis and observation of what occurs in the following stages after sperm-egg fusion, that a single pronucleus is formed when the sperm-egg fusion occurs within 80 microns from the emission of the polar body too. So this is an example how apparently trivial or totally unnoticed elements, morphokinetic elements of fertilization, can be harnessed in order to study the origin of normal fertilization and try to, in case, propose a cautious clinical use of those abnormally fertilized oocytes. We also in recent times observed more extensively abnormal fertilization, 1PN and 3PN fertilization compared to 2PN, and observed some interesting features. For example, we, I think, demolished the previous widespread belief that the PN incidence is mostly due to retention of the second polar body after ICSI. So therefore, 3 PNs occurs when the second set of maternal chromosomes are not extruded and they generate a third pronucleus, simply because the second polar body is not emitted. But in fact, we found that the emission of the second polar body occurs in over 90% of 3PN zygotes. And there are implications for that. One is that this is very basic. Dissociation of cytokinesis may occur from karyokinesis. So we're completely different, occasionally, phenomena that should occur actually at the same time. And we have observed no possible mechanism of generation of aneuploidy, and maybe Antonio may be interested in that, in which segregation failure affects not one, not few, but all or most female chromosomes, which again is something of relatively recent concept. And again, by looking at 1PN zygotes, in particular PN size, we can extract more and more information and try to marginally improve our results in the clinic. In particular, it has been seen that the size of the pronuclei in 1PN zygotes can be different, maybe larger, maybe smaller. And an obvious question we may want to ask is whether this different size may reflect a different chromosome content, smaller, haploid, larger, maybe diploid. And in fact, there may be some preliminary information which suggests we'd encourage to have a further experience in this respect. This is a study published already a few years ago in which the pronuclear size of 1PN zygotes was assessed in terms of ability of relevant abnormally fertilized zygotes with respect to ability to develop to good quality blastocysts. As you can see here, until a certain size, say 700 micron square, the ability of those 1PNs to develop to blastocyst stage was very poor, if any. And of course, they also, for completeness, showed some data on 2PN zygotes. However, when they measured the PNs of 1PN zygotes having a larger nucleus and observed further development, they noticed that the quality and quantity of development to blastocysts was almost comparable, or virtually comparable to the 2PN controls. So that tells us that there is maybe a possibility, a system maybe by PGTA, through which time-lapse technology allowing precise measurement of PN size at specific time points can allow for an early or preliminary prediction of normality of 1PNs. And again, we can move on further down the line of this observation and mentioned a number of other situations in which time-lapse has been very useful, especially in combination with fluorescent microscopy. Tommaso Cavazza, who is actually a scientist from Bologna originally, was able to show that the so-called nuclear precursors bodies, which has these dots within the pronuclei, although morphologically difficult to track by static observation, when they are observed by time-lapse technology, can actually reflect the distribution and condensation of the chromatin when you compare, as you can appreciate in these different panels, transmission light microscopy, time-lapse, with fluorescent microscopy. And when you track appropriately by time-lapse those NPBs, you can therefore appreciate whether the chromatin, which is reflected in the position that those NPBs may be a cluster at the interpronuclear area or condensed or scattered. Why is this important? Because we now know that, through the same study, that in order for the first mitotic spindle to be able to capture correctly all the chromosomes of the zygotes after pronuclear breakdown, those NPBs and therefore the chromatin, which is signaled by NPBs, need to be clustered in the interpronuclear area. Other patterns in which NPBs and therefore chromatin are not formed or they are cluttered is expose the zygote to the inability of the mitotic spindle to capture all the chromosomes simply because they are more scattered at the moment of PN breakdown. Now, this story of the first mitotic cleavage is very interesting because the more we observe this stage of development, the more we find out how critical it is for the entire development. This study was published last year and again combined time-lapse technology with the fluorescent microscopy. And what the author did was to follow the actual movement of chromosomes after pronuclear breakdown. And they observed a number of situations in which chromosomes were not being captured by the mitotic spindle, as I shown before. And they came to some interesting conclusion. The first one was that the first mitosis, compared to the second, is much longer, much more stretched, much more extended in time. And that may mean something. And all the different stages, pro-metaphase, metaphase, etc., are extended. And this extension in times actually underlines a number of abnormalities of the first mitotic spindle, which can be, in some cases, multipolar. It can be bipolar, but with some lagging chromosome, etc., etc., etc. And the interesting thing to notice here is that the delay in the process of the first mitosis is reflected in abnormalities of the mitotic spindle and an increased risk of mitotic aneuploidies, as we've seen before with Chris. So there are a number of things that are coming nicely together from different disciplines, different observations, different points of view. And I think this is the beauty of this area of research. Another example, a little bit less widespread, if you like, because it relates to a rare mutation, but very relevant to what we are talking about. And the topic is relevant to a mutation in this PADI6 gene, which is a component of of a multi-protein structure which is placed beneath the oocyte surface. And this structure, which has been shown to be existing also in the human, is fundamental for the very first stages of human development. Now, when controls of this case report are shown in green and indicate how, in the absence of this mutation, usually early development occurs normally, with normal divisions from the zygote stage to the early stage of development, two-cell stage. But when patients present with this mutation, there is a very high frequency of abnormal cleavage, especially trichotomous cleavage or even more seriously cleavage into 4 cells. Now, therefore, we now can harness time-lapse microscopy with other disciplines, including genetic screening, to make sense of what we see in the laboratory. Why those early zygote, the early embryos, why zygote divide abnormally, give us such arrested embryos? There are many reasons for that, but one of those may be a compromised configuration of so-called maternal effect genes. And there are also occasional observations, which, however, are very instructive of how things can happen. You can perhaps appreciate the slit in the zona, which probably was generated accidentally. And I'm going to show you what happens in the next next several hours after ICSI. 2 zygotes, 2 PNs were generated, but during the first cleavage, the early embryos was split in 2. And actually, the 2 parts developed independently over an expected, more or less, time frame. And perhaps you can also notice that the 2 blastocysts forming from the splitting embryos, actually, they also have a inner cell mass. So they can presumably be used for treatment. So I think over the last several years, we have elaborated an emerging concept in which prevails the notion that the first cleavage is a very perilous breach across the early phases of embryo development for a number of reasons that we have discussed, we will continue to discuss during this meeting. But it's not just fertilization, which is, however, is a topic that I like particularly. There are a number of other stages in which time-lapse can be harnessed. For example, the transition from cleavage to blastocyst. And this is very spatial, not only because many embryos arrest, but also because it is the first manifestation of morphogenetic event. So in development in action. And of course, we would like to see a morula undergoing full compaction and forming a beautiful blastocyst. But we've seen over the last several years that those are parts of the embryo that fail to compact do that according to specific patterns. Other cells fail to be included in the compaction process from the beginning, or they initially can undergo compaction and only afterwards be excluded. Now, we know from recent work that both patterns of failed compaction affects, in fact, the majority of morulae. And these can affect one or more cells with obvious implication in terms of size of the defect. And whatever the case, exclusion and extrusion affects all the major clinical and laboratory parameters on which basis we measure our performance. So it is an important manifestation on embryo quality that would have gone unnoticed without time lapse. And in fact, these initial observations were confirmed independently, also in the bovine. So I would tend to believe that this is a confirmed mechanism throughout evolution. We can also, by time lapse and other modality of manipulation, monitor the tension of those cells that fail to compact. And we can observe that the apical tension does not reach an appropriate level of change in its value in cells that fail to compact. In other words, the tension of the apical part of those cells does not reach the desired level. And that is preliminary to the ability of the cell to compact. In other words, when the apical tension and the lateral tension between cells are not properly regulated, there is a failure in compaction. So we are starting to understand at least something about the biophysics of this process. What causes tensor failure? We don't know that, to be honest. But maybe aneuploidy could be one of the several players. Reinterpreting blastocyst is another aspect that has experienced an acceleration of the knowledge through time lapse. Now, initially from study of the EV group, we know that one of especially repeated contractions is not good news for the blastocyst. When this occurs, the blastocyst is clearly less competent to implant and more exposed to risk of aneuploidy, as shown also by our group based in Rome, Jena. Now, I hope I've been able to, with this example, which are not exhaustive, but I hope representative, to convince you that harnessing time-lapse technology, together with a number of other different technologies and platforms, live cell imaging, image analysis, particle image velocimetry, genetic screening, and now artificial intelligence, we can definitely make a significant progress in research and possibly innovation, clearly, ultimately in the interest of our patients. Thank you. I think.
So, are there questions? No questions?
Okay, I would like to make a very easy question to Dr. Ahlström. So an IVF lab without time-lapse technology today is just losing knowledge or is losing chances of success in terms of live birth rate?
I think it's too early to say. So I should confess, we've implemented time-lapse and we use it, and I have no intention of losing it in our laboratories. I think it's too early. I think AI is changing the platform again. I think it's moving forward. I think there's, you can never design a perfect RCT or design a perfect study that will really sort of say it will give an impact or it won't give an impact. So we just need more studies. And that sounds so terrible to say that, but I think there must be some, and you see the connection Giovanni's making with the collapse and the connections that they're making with Blastocyst morphology, and we've seen studies that make connections, but if we all can't make those connections, then it won't have a huge impact for everyone with selection and morphokinetics, and maybe AI can sort that out for us with this image analysis. But yeah, too early to say.
Thank you. No question for the audience? Professor Zuccotti.
It's actually for both of you. Given the importance that time-lapse is providing to our studies and to possible clinical applications, do you think that it's time now to sort of establish some rules for movies acquisitions, so that the acquisitions that are made in one lab can be compared with those that are made in a different lab in a different country?
It is an important point, but it's not an easy question to answer, because I'm just wondering whether we're still experiencing a creative chaos situation in which everything makes sense before what is important can be regulated. I think it depends on the type of application, because in what we really want to look at, because, for example, in the case of particle image velocimetry, therefore, monitoring of cytoplasmic particle movement may be important, for example, the definition of the quality or the image, for example. Not necessarily so when you want to look at time of cleavage, because those times are not excessively quick. So therefore, whether you assess those timings at 15 minutes interval or 20 really doesn't make much difference. So in final analysis, I think we need to be specific when we want to answer those questions. So in some cases, some kind of agreed rules would be beneficial.
Okay, so if there are any, just a short one, please, because we're finishing. Okay.
Yes, it's a question for Ahlström. You mentioned that your data set is about 10,000 images.
Sorry, I can't hear you.
Oh, sorry. You mentioned that your data set is about 10,000 images. We wanted to know if you, how you acquire this amount of images, if you implement some augmentations, technology, practice, or other things. It's related with the model, artificial intelligence
Which
model.
study are you thinking? They're related to the IDA score or the images from the IDA score, or?
The images that you related with your data set.
With my RCT? Sorry, I don't think I understand the question.
Well,
Maybe-
we can...
Okay, maybe you can talk
Can we
each
take it
other
afterwards? Is that okay?
afterward. Okay.
Yeah.
Okay,
take-
so thank
Okay,
you so
bye-bye.
much, everybody. We move to the next session. Thank you.